Mitochondria are the organelles that produce the ATP your body runs on chapdarn, the power plants of the cell. And creatine's relationship with mitochondrial function is deeper and more interesting than simply being a buffer for the ATP they produce. The phosphocreatine system and mitochondrial energy production are not parallel tracks. They're physically and functionally integrated. Here's the mechanism. Inside each muscle cell, creatine kinase, the enzyme that converts between creatine and phosphocreatine, is present in two locations. One pool of creatine kinase is associated with the inner mitochondrial membrane, positioned right where ATP exits the mitochondria. As ATP leaves the mitochondria, this mitochondria-associated creatine kinase immediately uses it to phosphorylate free creatine, converting it into phosphocreatine. The phosphocreatine then diffuses rapidly through the cytoplasm toward the myosin heads, the sites of ATP consumption and muscle contraction. At those sites, a second pool of creatine kinase converts phosphocreatine back into ATP right where it's needed. The free creatine released by that reaction diffuses back to the mitochondria to be rephosphorylated. This cycle, the phosphocreatine shuttle, is not a backup energy system or a reserve tank. It is the primary mechanism by which mitochondrial ATP production is coupled to contractile ATP consumption in actively working muscle. Without adequate creatine in this shuttle, the efficiency of ATP delivery from mitochondria to myosin heads decreases. The diffusion of ATP itself from mitochondria to contraction sites is slower than the diffusion of phosphocreatine, because phosphocreatine is a smaller, more diffusible molecule in the cellular matrix. The shuttle effectively extends the reach of mitochondrial ATP production by translating it into a faster diffusing carrier molecule. Research on mitochondrial function and creatine has also explored creatine's role in mitochondrial biogenesis, the process by which cells build new mitochondria in response to training demands. Some evidence suggests that creatine supplementation, in conjunction with exercise, slightly enhances the mitochondrial biogenesis signal. The proposed mechanism is that better maintenance of ATP levels during exercise. Through the phosphocreatine buffer, reduces the AMPK activation that would otherwise signal energy stress and trigger mitochondrial biogenesis. This sounds counterintuitive. Lower AMPK means less biogenesis signal, but the actual evidence in exercising humans is mixed, and the practical relevance for most athletes is modest. What is clear is that creatine and mitochondria are not separate systems. The phosphocreatine shuttle requires healthy, well-functioning mitochondria to work. The rephosphorylation of creatine at the mitochondrial membrane is powered by mitochondrial ATP production. And healthy mitochondria function more efficiently when they have adequate creatine to operate the shuttle. Because without the shuttle, they're trying to do the same energy transfer over longer diffusion distances with a less mobile carrier molecule. Supporting both mitochondrial health. Through aerobic training, adequate micronutrient intake, and anti-inflammatory dietary practices, and creatine saturation through consistent supplementation, is the way to maximize this integrated energy system. The two are complementary, not redundant. The brain-mitochondria connection extends the relevance of creatine's mitochondrial effects well beyond skeletal muscle. Neurons are among the most energetically demanding cells in the body. Unlike muscle cells, which can switch between aerobic and anaerobic energy production, neurons are almost entirely dependent on aerobic mitochondrial ATP production for their function. When mitochondrial function in neurons declines, through aging, disease, or acute injury, neuronal energy supply drops, and the phosphocreatine buffer becomes critically important as a short-term bridge to maintain ATP levels. In Alzheimer's disease, Parkinson's disease, Huntington's disease, and ALS, mitochondrial dysfunction is a consistent early feature. The progressive decline in brain energy metabolism precedes the onset of symptomatic neurodegeneration in several of these conditions. Creatine supplementation has been investigated in each of these diseases as a potential neuroprotective agent, specifically because of its ability to buffer the ATP shortfalls that result from mitochondrial dysfunction. The clinical trial results in these diseases are mixed, and creatine is not an established treatment for any of them. But the mechanistic rationale is compelling, and the ongoing research in this area represents one of the most important emerging dimensions of creatine science beyond athletic performance.